In this lecture, you're going to learn about the histological structure of bone marrow and the process of hematopoiesis. Sometimes the word hematopoiesis is also written and pronounced hemopoiesis. So I might use both terms intermittently throughout this lecture, but understand that hematopoiesis and hemopoiesis are the same process. Well, in this lecture, I'm just going to briefly cover some of the major lineages or major processes whereby stem cells can give rise to progenitor cells, and each of those populations of progenitor cells can then give rise to the blood cells. And in this lecture, I'm only going to cover the lineage that gives rise to the erythrocytes, red blood cells, and as an example of the granulocytes, I'm going to look at the lineage that gives rise to the neutrophil. So in other words, I'm going to describe the events called erythropoiesis, manufacturing or giving rise to red blood cells, and granulopoiesis, giving rise to the granulocytes, of which only the neutrophil is going to be the example I'm going to use. Now, bone marrow has a number of functions. It's an enormous tissue. It is important to give rise to all the blood cells. It produces all the blood cells and then releases those blood cells into the blood once they're in a mature functional form. Blood sinusoids, or blood capillaries, special vessels in the bone marrow, are very important to maintain a barrier between the circulation containing normal blood cells and all the cells that are undergoing all their training and development. I'm not going to talk about the blood sinusoids in this lecture, but I just want to stress that they do form a very important barrier so that external factors don't interfere with the process of hematopoiesis and the training of all these different blood cells to be able to do their job once they're released into the circulation. I'll talk about blood sinusoids in more detail when we look at the cardiovascular system and other systems. Hematopoiesis includes formation of the red blood cells, the white blood cells, and also the thrombocytes or platelets. Let's now look at some of the details of hematopoiesis. Firstly, it's important to know that all the blood cells that circulate in the blood have a very limited lifespan. Red blood cells only have a lifespan of 120 days. Neutrophils even less. So it's important that we have a tissue that continually renews all these blood cells. And that's the process of hematopoiesis, and it occurs in the bone marrow. The original stem cell that gives rise to all the lineages is called the hematopoietic stem cell. It's a pluripotent stem cell. It's obviously a very important cell. It sits in the bone marrow, it divides over and over again, and it gives rise to the progenitor cells, which in turn go through all the process of providing lineages leading to the production of all the different blood cell types. So it's important that this hematopoietic stem cell divides continually to provide these progenitor stem cells as well, because they're lost during the process of their differentiation into the final blood cell types. There are a number of different colonies that arise from these hematopoietic stem cells. One is the common lymphoid progenitor cell types that give rise to the lymphocytes, and I'm not going to talk about them in this lecture. Then there are the common myeloid progenitor cells, of which have different progenitor cell populations that are then influenced by a number of factors that is not the scope of this lecture. a number of factors that then stimulate these lineages to go through various processes of developing into the various cell types. The erythrocytes, the white blood cells, megakaryocytes, and the platelets. So in this lecture, I'm going to concentrate on the lineage that's derived from the megakaryocyte, erythrocyte progenitor cell. And then I'm going to talk about the lineage that gives rise to the neutrophil, the lineage that is derived from a granulocyte, monocyte progenitor cell. But appreciate there are different colonies of these progenitor cells, and as I've said, they're influenced by a number of factors, some of which I've listed on this slide. Well, let's first of all look at erythropoiesis, the lineage that gives rise to our red blood cells, those very important cells that transport oxygen and carbon dioxide throughout the body within blood vessels. Well, it's important to understand that there are a number of stages in erythropoiesis. And again, let me stress, they arise from the megakaryocyte or erythrocyte progenitor cell. Have a look at this slide on the right-hand side. You know, one of the most fascinating things when you study histology is you see some very, very pretty slides, very pretty stains, very pretty demonstrations of tissue. I think bone marrow is one of the prettiest tissues to look at using standard stains under the light microscope. Well, when you look at this slide, you can see some red blood cells that have maintained their mature red blood cells. All those small pink, reddish or orange stained structures are erythrocytes. They've matured, they're circulating in the bone marrow, and they're leaving the bone marrow to enter into the general circulation. They arise from the proerythroblast. This is the very first stage of the progenitor cell then starting to form what will be the final erythrocyte. These proerythroblasts are large cells. They occupy a diameter of up to 20 microns. So they are big cells. And one thing to bear in mind is, particularly when you look at lineages of this cell and the neutrophil later on, is have a think about what the end product is going to be. The end product is going to be the red blood cells that you see also in this image. They're much smaller cells. They're only about 6 to 8 microns in diameter. So when the progenitor cell gives rise to a proerythroblast, it's a large cell that is eventually going to go through the differentiation process and finally end up being just a little package of hemoglobin. And that's essentially what a red blood cell is. So when you see these large cells, they represent the very busy activity of these cells. And I'll refer to that as we go through this lineage. The proerythroblast has a mild amount of basophilia. Suddenly the progenitor cell has given rise to the proerythroblast. It's going to now start going through the process of making hemoglobin. But initially, it just has a mild amount of basophilia. Well, the next stage in the process of forming a red blood cell is differentiating into what we call the basophilic erythroblast. And that is shown on this particular cell here. And basically, if you look very carefully in the cytoplasm, there is stronger basophilia than you saw previously in the proerythroblast. And then again, as I said earlier, think back as to what the destination of this cell is going to be. It's going to be a red blood cell. It's going to be containing a whole lot of hemoglobin, the protein carrying oxygen. So initially, back in the early cell stages, you would expect to have a cell that develops an enormous amount of rough or granular endoplasmic reticulum. to make that protein hemoglobin. So at some stage, and that's the stage you see here, the cell has accumulated all this endoplasmic reticulum, which takes up the basophilic stain. So here is an indication now that this cell has been activated to start making hemoglobin. It has the factory now to make that hemoglobin. Well, the next stage is the polychromatophilic erythroblast, shown here. Notice the cell looks small there, even though they can have a different range of diameters, ranging from 10 to 12 up to about 20. So there is a variation in their size. But have a look at the cytoplasm. It's sort of a lilac colour. It's lighter. And it's because this cell has now gone on to the stage where, having got the factory to make hemoglobin, and therefore basophilic, it actually manufactures the hemoglobin. And hemoglobin, being a protein, takes up the eosinophilic staining. So you have a combination then of a bluish tinge representing the stain of the endoplasmic reticulum, and you have the reddish eosinophilic type tinge related to the production and accumulation of hemoglobin. And therefore it looks a lilac type colour. We call it polychromatophilic because polychromatic means many colours. The cell then passes to the stage where we call an orthochromatophilic erythroblast, commonly called a normoblast. Orthochromatophilic means it's only one colour. And if you look at the cell, it's got essentially a reddish tinge around the cytoplasm. The nucleus is very, very condensed now because the cell has now become a lot smaller. On its final destination to be a red blood cell, only 6 to 8 microns in diameter. But note the colour of the cytoplasm. It's now more eosinophilic or reddish, to do with the accumulation of the protein hemoglobin. And here's a normoblast shown here, where you can't see the cytoplasm. It happens to be in another dimension, but it again is to stress the fact that these nuclei are very, very condensed. And that's a good way of recognising these normoblasts or these orthochromatophilic erythroblasts in bone marrow. Finally, that nucleus is going to be extruded from the cell because, as you recall, the erythrocyte has no nucleus. It is just the package of hemoglobin. Well, it then goes through the stage of being a polychromatophilic erythrocyte again. And finally it goes on its final lineage differentiation to be a fully functional mature erythrocyte. And the reason why it's now referred to as a polychromatophilic erythrocyte, or sometimes we refer this as being a reticulocyte, is because as the cell gets smaller, the cell still retains a little bit of the factory that synthesizes hemoglobin. The cell has lost its nucleus, which is why we call it a reticulocyte, as opposed to a normoblast you saw earlier. But because it's got a lot smaller, as I mentioned a moment ago, it has hemoglobin in it, which stains pink or reddish because it's a protein, but it also has still traces of the protein factory and therefore traces of a blue stain, and that therefore gives the appearance of these cells as being polychromatic or a lilac-y colour. And finally, that erythrocyte is now fully mature and it's released into the circulation. It's nice and rounded, it's got a biconcave disc shape, and now it's full of hemoglobin. So, in summary then, it's important that you appreciate the lineage that I've described, but the main real point I think to understand is that you are going to end up with a cell that is full of hemoglobin, the protein. You start with a progenitor cell that has to then create lineages that first of all develop a factory to make the hemoglobin, the endoplasmic reticulum, that stains very strongly basophilic, and then as the cell moves through the process of making hemoglobin, then there is a combination of coloration you see in the cytoplasm. And then finally, as you get more and more hemoglobin produced, then the cell becomes more pink stained. So remember the destination of the cell and remember the process involved. And I think it helps you to think about the sort of staining that you see in this lineage. It is very hard to identify these cell types in bone marrow, particularly using some stains that just don't happen to work as good as we would like. But what I hope I've given here is some demonstration at least of the lineage giving rise to the erythroblast, the erythrocyte from the proerythroblast. Let's look at thrombopoiesis, or the formation of the platelet. Here is a huge cell. This is the megakaryocyte. It's multi-lobed. It's often has many, many, many different components to the nucleus. It's a huge cell, up to about 70 microns or even bigger. But what happens is that the cell membrane around that megakaryocyte invaginates, and invaginates a number of times into the internal components of the cell. And those invaginations come together and join up, and therefore part of the cytoplasm is cleaved or broken off and released into the bone marrow and then into the blood system, the plasma, and the red blood cells within the vascular network. And that is a platelet or a thrombocyte. And as I explained in a lecture on blood, these thrombocytes have very, very important functions, particularly in helping reduce the leakage of fluid during damaged parts of the wall of a blood vessel. Well, now let's look at granulopoiesis, the formation of the granulocytes. Here you see another image of bone marrow. And what we want to concentrate on now is the granulocyte monocyte progenitor cell lineage, the lineage that's going to give rise to the granulocytes and also the monocytes. But as I mentioned earlier, I'm only going to look at the formation of the neutrophil. Well, the first cell that starts off in this lineage is the myeloblast. It's a large cell again, it's about 20 microns in diameter, and it's got a large euchromatic nucleus. Now, when a blast cell forms, when cells often revert to being a blast type cell, then they're very busy cells. They're manufacturing components. And we'll see that as we look at cells, particularly with the immune system and other tissues later on. Because they're very busy cells, therefore, they have a very euchromatic nucleus rather than a more heterochromatic nucleus, which reflects usually that they're not so active, they're resting. Well, now let's look at the next cell type in the pathway. Let's see where this myeloblast then moves into or differentiates into. What sort of a cell is it and what does it look like? Well, it differentiates into the promyelocyte. And this promyelocyte contains or starts to generate the factory that's going to make the azurophilic or primary granules. I say the word primary here because these granules appear in all the granulocytes, the neutrophils, the eosinophils and the basophils. But you will recall, if you have knowledge of these three different granulocytes, that these granules are quite different in their content as they differentiate into their respective cell types. So initially, This part of the lineage starts to accumulate these granules, and therefore they stain with the Asia dyes, but they're yet to go through the differentiation into acquiring granules and the contents of those granules that are specific for the cell type. So this is a general cell. It's yet to really determine which direction it goes into. Does it become a neutrophil, or a basophil, or an eosinophil? The next stage that this cell then passes into is the myelocyte. In those cells, the specific granules will emerge. Again, those specific granules relate to the different roles that these granulocytes will have. For instance, the specific granule in a neutrophil contains antimicrobial substances or enzymes like collagenase that help digest the microbes and the bacteria that they're going to destroy and then take into their cell cytoplasm. Whereas the eosinophils and the basophils will have specific granules for their particular role or purpose. So these myelocytes, again, are quite large cells, and again, if you look carefully in the cytoplasm, you see evidence of a granular structure, but you really can't distinguish what type of granules they are at this early stage. Well, the cell then passes into the metamyelocyte stage. And the metamyelocyte stage is a very important stage of development because it's at this stage that the cell can no longer divide. Hence the little asterisk I've put next to the term metamyelocyte. But what happens is that the nucleus becomes kidney-shaped. And sometimes you can see that kidney-shaped nucleus when you look at these cells when they're orientated in a way in which they appear when viewed with the light microscope. Sometimes if that cell was rotated around because you're looking at a smear of bone marrow, you wouldn't see the kidney-shaped nucleus, so it would be difficult in this instance to then identify that cell. But that is one of the characteristic features of the metamyelocyte, and let me stress again that this is the stage that the cell can no longer further divide. Well, in the case of the neutrophil, that nucleus that was originally a kidney-shaped or a bean-shaped then develops further, and we call it a band or a stab cell. Have a look at the nucleus and you see that it's a horseshoe-shaped or a U-shaped. This is an indication that the cell nucleus is going through the stage where it's actually going to be the polymorphonuclear appearance that you see characteristic of the neutrophil. And at the same time that the nucleus is going through a differentiation process, two other things happen. Firstly, the granules start to become specific for that particular cell type. Because it's no longer dividing, it's concentrating on being the lineage to which it's destined to be. It's concentrating on creating the factory inside the cell, within the different granules, to fulfill the role of that cell type. In this case, it's a neutrophil. And finally, that neutrophil, or that nucleus, will go through a stage where it becomes, as I said before, polymorphonucleus, or having many forms, many shapes. So, again, I hope you have some understanding of the various lineages whereby the myeloblast can give rise to granulocytes, and concentrate again on the actual finished product of these cell types. The neutrophil, the eosinophil and the basophil each have specific granules within them, containing different components that serve the role of these cells. So as the cells develop through their lineage, first of all, they go through a process of just generally producing granules. And it's only after the metamyelocyte stage, where they can no longer divide, that the cell then, destined to become one of those three types, goes through the process of forming the specific contents of those granules. So now I hope you understand a little bit about bone marrow, and thank you for listening.